I've selected internal Mac, but it's not coming on. Usually when I push it, the screen comes right up. Yep. I have my slides loaded on that computer. Oh, so what did you do?
I just turned it off.
Okay, so this is a new system, and then you have to touch it before, otherwise you get that black. No, it always opened before to the desktop. I never got that black screen before, so that's something new. Okay, thank you. Okay, I think this is working. We're ready to go. What we have here, as I said, we're going to go through each unit, and it doesn't necessarily go by lectures. So we are in the minute here. We are in the middle of Unit 1, Regulation of Metabolic Pathways. For the viewing audiences and for those here, we'll be starting with PowerPoint 1.18. Okay, so I was approached afterwards about just mechanisms of study. The organization of this section is coming in through the PowerPoints. I will not necessarily say everything on the PowerPoints, but that's information that you should know and understand. The notes that you get where your reading assignments are kind of supplementary in case you want more information, as is the book. I mean, if you can answer the problem set questions and follow it from just the information on the PowerPoints, that's fine. Now remember, I could say something like understand glycolysis, so that just means that's an important pathway. It's not all the details, but it's helping you with this process that I said was important, which is overview. So I was in the middle of regulatory mechanisms, so I just wanted to start with this. We can control it at all levels. I did go through this. These are the things you learned about controlling enzyme activity in Part 1. These are the things that Dr. Taub will talk to you about in Part 3, but we will talk here a bit about protein degradation, but that's in the second part of this exam. The compartmentalization is something that's most relevant to what we do and discussed here. When you want to integrate all of these various ways of controlling metabolism, which remember has to be done very quickly, hormonal regulation integrates this metabolic activity and how it is interacting with both carbohydrates, fats, overall metabolisms will be a constant theme of this section of the course. So each of the types I talked about before, this is a reiteration of where they are discussed. And the problem of compartmentalization is affecting metabolic rates. And what you learned about in the first part with membranes is that they're basically impermeable. What you need to be thinking about in terms of getting things across a membrane is it is impermeable to everything. By itself, a lipid bilayer is impermeable to protons. So if protons don't get across, small metabolites aren't going to get across. You learned about active and passive diffusion. So for every time we want to get something from one compartment to another, we're going to have either an active transport system or a passive, a facilitated diffusion. And the problem with a facilitated diffusion is it will only transport with concentration gradients. So if we want to do something against the concentration gradient, we have to provide energy, and that will happen. So if you have your cell full of something, and it's in a certain compartment, and it's in the mitochondria, and you need it in the cytoplasm, you might as well not have it. So therefore, this compartmentalization is extremely important, and we'll be talking about that. That is compartmentalization is a type of regulation. All right, so we want to control metabolism. If you're going to have something that's going to be a quick on and off, you're going to have things going in both directions. The winner in the game of molecular evolution turns out to be phosphorylation and dephosphorylation. That's the major mechanism that covalent mechanism that we're going to be talking about in this section of the course. So to help you understand what you see there, if a protein phosphatase, remember I said people are bored with naming or name things in a boring way. If something is called protein phosphatase Y, it will cleave a phosphate from protein Y. So the name is telling you what that does. Now, we don't, for phosphorylation, it's kinase. When you see kinase, that means it's going to put on a phosphate. So protein kinase X adds a phosphate to protein X. So again, if a protein is phosphorylated, it's going to be a kinase. If it's dephosphorylated, the phosphate's taken off. The enzyme that does this is a phosphatase. So if you have something that I'm talking about, or anyone else, in a signal transduction pathway, and we say it's phosphorylated, then right away you know there's got to be another enzyme there that dephosphorylates. And what you'll see as we go through this section of the course, sometimes you have a random, what I think of as a random kinase or phosphatase that has a substrate specificity that will act on a variety of different proteins. And sometimes you have one that are specific for just the protein that we are studying. This you can take to the bank. If the phosphorylated form of the enzyme is active, then the dephosphorylated form is inactive, and vice versa. So that means just what it says. So all phosphorylated forms are not active. It's just for certain cases you might have the reverse of this. You could have the phosphorylated form being inactive, and then you know the dephosphorylated form is active. So in other words, there's whatever is existing in a certain condition, the opposite will have the opposite effect on metabolism. How this is important in this section, Dr. Ponticelli, I think as the specific example, gave you the cyclic AMP-dependent protein kinase. You guys are supposed to be all primed to know about cyclic AMP. Did that happen? Well, let the viewing audience know that they're not quite sure about whether it happens or not. But everyone seems, I'll go over it again, but you seem to, at least it's a familiar term. So when we're controlling, when we have hormones that act, they might start phosphorylation, right? That's all they can do. A hormone can do a certain thing. Then what happens is that if you want to what you'll see in this section, let's say glycogen metabolism. You have key enzymes involved in both the synthesis of glycogen and the breakdown of glycogen. If phosphorylation is only the thing that your enzyme can do, and if all enzymes were, let's say, always active when phosphorylated, then if you phosphorylated both key enzymes, the one involved in breaking it and the one involved in making it, that makes no metabolic sense. In other words, you want the phosphorylated form of one to be active, let's say, to make it, and the one to be inactive to break it down. So you can't tell whether the phosphorylated form is going to be active or inactive off the top of your head without considering a lot of other things. But once you know one of them, if you know the dephosphorylated form is inactive, then you automatically know the phosphorylated form of that enzyme will be active. Okay, so do you see how that goes? They're always opposite, but you can't a priori figure out which the first one is going to be. Now, as I said, this was the winner in molecular evolution. We also have some runners-up. We're focusing on phosphate, and when I talk about phosphorylation, this is just a diagram. Your energy, the terminal phosphate of ATP is being added to your enzyme to form the phosphorylated form. Now, there are some cases where an adenylate moiety is added, and here you see that adenylate added. And then you actually sometimes put, for the whole 9 yards, an ADP ribosyl moiety. And we don't actually discuss any of these. But again, this is from the 3rd edition. I think if you're in the 4th edition, something new that has turned out to be really important is methylation. It's a covalent modification of enzymes, and its regulation has been really, the importance of it has come up in the last decade or so. And it's something we see a lot in what we call post-translational modification that Dr. Taub will be telling you about. So, yep. Yes? Well, well, the idea is, I'm not going to show you the structure. On the exam, there are going to be some questions, as I said, where instead of giving you a name, I'll give you a structure. And clearly, it'll be a structure that came from this section of the course. So, for instance, if I were to say this compound is involved in is a covalent modification, true or false, that's a legitimate question. But it's recognized, okay? It's not be able to draw it. Whoa, I lost something here. Okay, so compartmentalization. It's really a way of keeping information sorted, and so what's in one is not available to another one, and it's basically a real physical separation by these impermeable membranes that I was talking about, and it's important. It keeps apart metabolites that may be involved in redundant cycles. If we have a pathway and we're going to utilize the same enzymes, so you have one side of the pathway breaking it down, catabolism, the other side building it up, anabolism. Well, you might want to mess around with the concentration of the substrates. And that's, again, the fluxes along those pathways is the same thing. It allows us to concentrate metabolites if we want something to happen in a particular compartment. So if we want something to happen in one compartment and not in another, we can make sure it has more of a particular metabolite, and you'll see how that's important with the thermodynamics. And it also can concentrate enzymes in a locale to increase efficiency and also to prevent diffusion. And you've already had, I don't know in what depth, but Dr. Ponticelli did it, but you've learned about the enzymes that break down proteins. You've learned about protease, chymotrypsin and trypsin. Now, when you think about it, when chymotrypsin is made, why doesn't it just destroy the whole cell? So the reason for that is zymogens, is that chymotrypsin is not made in an active form. The thing that activates it is actually present in another compartment. And both of them, as we'll learn about in digestion in the second part of metabolism, when they both are released together, then those enzymes are activated. That's a mechanism that compartmentalization is used to keep the body from digesting itself. And we have other examples of that coming up. So remember, what you're seeing from what I'm saying is the first section of this course, from a metabolic point of view, introduces a whole bunch of concepts that we're now going to grab and use for regulation and see what they're used for in the body. Now, there are a lot of places in the cell that you have to worry about. If you've had a cell bio course, there are lots of compartments. So to simplify things for this course, we're focusing on the cytoplasm, which is the gray stuff back here, and the permeability barrier for that is the plasma membrane. That's what's differentiating our cell from what's in the outer portion of the cell. Then what we're dealing with is mitochondria. So we'll be spending time talking about things happening inside the mitochondria, outside the mitochondria, and what's outside the cell. There are various processes. This is only a few weeks of metabolism that we're not discussing, where whether things are present in the Golgi or the ER becomes critical. But again, this is an introductory course to introduce you to concepts. So the concept of compartmentalization is stressed here, by comparing the cytoplasm with its external plasma membrane to the mitochondria. Those are the two compartments we'll be focusing on. Now we get to thermodynamics. As again, nobody's violating anything. We have to obey the laws of physics. So it's the branch of physics concerned with energy exchange, and our first law is that our delta E, or our energy, is the heat released minus the work done. Now, when we're talking about metabolism, we're talking about chemical reactions. Now, we don't have any volume changes. I mean, unless you're eating something that's not food, you don't really think volume is changing when you're eating. And so our P delta V, or our delta work term, is 0. So this equation gets reduced to our energy is equal to the heat released somehow. That's all we have to worry about in metabolism. So now, how's the best way of doing this? If we want the total oxidation of something, the complete breakdown, then we use that calorimetric bomb that I was talking about, and we can get the total oxidation of any substance. Here it is. So you just go, I don't know if you've done it in chemistry, but what we're going to do, striving in this next few weeks, is to do the complete oxidation of glucose, having to add, so when I say oxidation, we have to add oxygen, and the end product is CO2 and water. So you'll hear me say a lot, the complete oxidation of glucose to CO2 and water. When we get partially through this, we've got it to CO2, but we have to then get it to the water with the respiration. So the total energy released is minus 2,816 kilojoule per molecule in a calorimeter. Calorimeter I can rely on. That's true. So therefore, that's one of these facts that we know. Now, when we talk about what's released from metabolism, we're making assumptions about what's happening for these different reactions, and we're not as sure about that. But we certainly would know that if we got 100%, we would be winding up here with 2,800 kilojoules. Now, again, most metabolic reactions are constant pressure. So the heat exchange in this condition is our enthalpy, which is our delta E plus our P delta V, and you know that's one's going to go to 0. So what we deal with is, so now we've wiped out the first law and everyone's still alive. We have to worry about the second law. And here it incorporates the natural tensity of any isolated thermodynamic system, of which you could be considered one maybe, to always evolve towards a state with less organization or more randomness. So the delta G, which is the G of the products minus the G of the reactions, will come from effects on enthalpy or T delta S. Now we know, because we can do the direction, that for a reaction to be spontaneous, our delta G has to be negative. So if you look at this reaction, two things could happen. We could be releasing heat. You can see how that's going to make our delta G negative if we have minus delta H here. Or if it's minus T delta S, we could increase randomness. So I mentioned this before, we're talking about converting our fuel to us. So how many of you had breakfast? Oh, only 80%. You know, you really should have breakfast. It's an important thing. I mean, but you know this. Okay, so for those of you who didn't have breakfast, at some point you did have breakfast, so you might have had something, I don't know, toast, congee, eggs, or whatever. So think about that breakfast and think about you. And which is more organized, you or the toast? Okay, how many people think the toast is more organized? Oh, a few. How many people think you are more organized? Okay, let the viewing audience know it was 3-to-1 people to toast. Toast is basically a polymer of carbohydrate, whereas you, as you saw from that diagram of the cell, have a lot of different things going, and even after that cell is made, there's other levels of organization at the tissue. So from an order of what we would describe as randomness, the toast is more random than you. I mean, by converting your food to toast, you're not increasing the order, the delta S here. So what's going on here, to get our delta G, we're going to have to be releasing our delta H, okay? Now, I'm not going to be taking questions here for all the time through class because we have a lot to discuss. Unless if someone has something with a mistake that they see right away, let me know or come up after class. Okay, so now what we have here is another thing about metabolism illustrating life is if we're saying we're not going to do it with our entropy term, we're going to be releasing heat. So how many people have eaten a meal and felt warmer? Few. How many people, another way that that's interchangeable with using energy in addition to eating is exercise. How many people have exercised and felt warmer? Let the viewing audience know that we're back up to 80%. So basically, when you do something like that and you're releasing the heat, what you're doing is basically, that's showing that you're not violating the laws of thermodynamics. And that's what we're focusing on here is heat. And so, okay. Now, how do we determine the delta G? Again, you've had this in the first part. The standard Gibbs free energy, if we have a reaction A plus B going to C plus D, you can have an equilibrium constant where we have the information for the reactants over the products, and we get our standard free energy. So our actual free energy, in other words, what we're really observing, is our standard free energy that we can get from the isolated things, plus RT ln K, or the ln of this equilibrium constant, and these are the temperature and a constant, okay? Now, what actually happens in a body is that we can sometimes calculate our delta G on the basis of volts. And here we have to have delta G minus NF delta E. Again, I think you've seen this in the first part, which is the number of electrons transferred times the Faraday times delta E. This becomes very important in the second part of metabolism. And what you need to remember, we want to go for a negative delta G to have something positive. So since our delta G is equal to the opposite of a delta E, that means we're going to be increasing our delta E to get that minus delta G. So therefore, it matters what you're expecting, whether you calculate things using this equation or you calculate with concentrations. Now, the body has a lot of different things going on. And what we're really interested in, and the textbooks, they can change what they call them, or the symbols, unfortunately. But there are three symbols that you have to worry about. This is given by the concentrations present at that time and determines whether or not, again, the reaction is spontaneous. So G0 is the fixed value that the physicists have come up with, and that's 1 molar. And 1 molar at certain temperature Kelvin and 1 atmosphere pressure. And that pretty much is okay. But we have here, except for 1 molar, but delta G0 is the fixed value for biological conditions, because you'll learn we care a lot about pH. And this is done at pH. And it can be calculated for 25 degrees centigrade and different concentrations. So you as a biologist might be going to different tables for certain reactions that are given for the standard conditions and converting them to the delta G at pH, or you might be actually just measuring what's there to get your delta G that way. Now, you can modify this if you want to, because we want things to be spontaneous. You can change the concentrations of reactants and products. You can change the pH. Or what really happens a lot is, as I spoke, you can couple an unfavorable reaction with a very favorable one, pull out some ATP, and the reaction will occur. So cell metabolism operates in a non-equilibrium condition. Everything is maintained at steady state. Now, that you also prove to yourself by the fact that heat was given off. The difference in an equilibrium condition from a thermodynamic point of view is a closed system. It doesn't interact with the environment at all. We are not closed systems. We're constantly giving up and taking in heat, and that means that we operate at steady state and not equilibrium. We can be approaching equilibrium constants and things like that, but we're really in a steady state. Okay, so how do we do this? This is the tricks of the trade. Here you have falling rocks, but living where we are, falling water is probably more likely what you see as the water goes over Niagara Falls. All this energy gets wasted if there's nothing to save it or to get it at the bottom. And one way you can do this is if you put in a turbine, which we have plenty of at Niagara Falls, and in this example, the rocks coming down lift the bucket, and then the bucket can go somewhere and do further work, the water that was in there. So this is a simple model done with potential, energy, and gravity, but the same thing happens with our chemical potentials. So what does that mean when we look at different reactions? If we have a reaction where A goes to C plus E, and the delta G is plus 5 kilocalories per mole, it isn't going to happen. But if we have another reaction, E plus B goes to D, and the delta G is minus 8 kilocalories, that will happen, right? That's spontaneous. The delta G is negative. Now, if you add these two up, you see what you wind up with is A plus B goes to C plus D, and you would add up the delta G's, and you could see that the delta G for this coupled reaction is minus 3 kcals per molecule. It will happen. So this goes on all the time, and this is something you should know about. If you look at your problem set, which is an old exam, you are responsible for being able to realize this and to figure out if there are questions saying, is this going to be a spontaneous reaction? And it gives you information and various delta G's, and you add them up, and then you figure out whether it'll be spontaneous. Now, if I had written this reaction as C plus E goes to A, this delta G would be reversed. Remember, because this is part of it, this would be minus 5. So when you do these types of problems, and you will be doing them on your problem set and exam, you need to make sure you have the reaction in the right direction for how the delta G was given. If the delta G was given this way and you needed it the other way, then you would just have to flip this sign. So that's something you do need to know. Now, thermodynamics versus kinetics. What we have from a thermodynamic point of view is whether it actually will occur. The kinetics is sort of telling us the rate. Again, this is from you've seen this in the first section, A plus B going to C plus D. We have our equilibrium constant that is governed by these rates. And again, our delta G can be gotten from the equilibrium constant that way. Now, this is a standard energy diagram, and you can see that you sometimes have a high rate constant in one direction, and without an enzyme, you've got to essentially build this up. So what an enzyme can do is it can lower this, and therefore you're more likely to have it happen, okay, because you've lowered the activation energy. Now, you haven't essentially, we're looking at what happens in the beginning and the end. The reaction would occur. It was going to be spontaneous, so the enzyme doesn't make that, doesn't say, well, something that was thermodynamically not going to happen will happen. But what you also see, this rate from a practical point of view, I mean, think about it. If a reaction is going to happen, and it's going to take 1 billion years before it actually happens, it's not that useful to you. So what we see is that even when I will refer to a reaction and I say essentially irreversible, or it's not going to go in that direction, I mean under the given circumstances in the cell, temperature, concentrations, and pH, the rate constants are so organized that it takes too long. So from our point of view, it's an irreversible reaction. But it doesn't mean that from a thermodynamic point of view, that it's totally irreversible or having a positive delta G. Okay, so what are the important parts about this that we have to know? Delta G values are additive and pathway independent. That's because it's a state function. So that means if I'm giving you from A to B, I gave you a pathway with B and C, but if you have a pathway involving different intermediates and know the value from A to E, that delta G will be the same for whatever path you use to get there. It's pathway independent. They take place, metabolic reactions are taking place at steady state rather than equilibrium. And the directionality is established by our favorable energetics by a few key reactions. So if delta G is less than 0, and this is most often due to the release of heat, it's irreversible going in our direction. Reaction rates are controlled by enzymes, and they proceed at very low rates in the absence of a catalog, of this catalog usually. Okay, so ATP, which you've seen before, is our chemical currency. Our high energy bonds are the last two phosphates. So we have two high energy bonds, and what we give out is 33 kilojoules per molecule. Now this last bond, which is only 14, is considered low energy. You can actually convert an AMP molecule here and an ATP molecule here to two ADPs. The enzyme that does that is adenylate kinase. What's important for us is when we're thinking out the energy charge, we want to know how many of our adenine nucleotides are in a high energy condition and how many are in a low energy condition. So here's our energy charge of the cell. And this equation, and this half ADP comes from the action of this kinase, of the enzyme I just spoke about. So if you have ATP plus half the ADPs, this is going to be how many of them are ATP. And then you want the fraction of the total adenine nucleate pool, which is ATP plus ADP plus AMP. And that is referred to as energy charge. What we have is, and you can take limits, as the energy charge goes to 1, this limit is approached as the ATP is much, much greater than the concentrations of anything else. Your energy charge will go to 0 when the ATP is much lower. That's simple math. So you have large amounts of ATP, and mostly ATP in the adenine nucleotide pool, and your ATP utilizing reactions will occur. That's up here when your energy charge is close to 1. In the other way, you have small amounts of ATP. That's the current currency for the cell. So clearly you want to make more of it. You've got mostly AMP, so you want the ATP synthesis reactions, which are up here to be done. Now notice that as you get for different adenylate energy charges, you'll wind up having different ratios. So as you go from 0 to 1, your synthesizing reactions drop. As you go from 0 to 1, your utilizing reactions increase. Now, this is a key regulatory point. So clearly, you wouldn't want cellular energy charge to be varying in this range, because then there will be no change. And what actually happens, because you can measure this, is the energy of the cell is sort of up here around 0.9. And it can vary a little. And it's varying over where the crossover point is for the reactions. So that means as this varies, you can switch from ATP utilizing reactions to ATP synthesizing reactions. And that's an important thing for metabolism. So I think, let me stop. Okay, we just have here the enzymes that are involved. So I'm going to stop here to give you your 2-minute break. So this is where I want you to think about what Dr. Ponticelli told you. Whoops. Okay, so you learned about, I reviewed the FAST. You learned about them in the first section, how enzyme mechanisms can be controlled. And I just talked about energy charge. So I want you to take the 2 minutes, either think about this or what a government shutdown means to you, or talk about whatever you might like, and then I'll come in with the answer about what was taught in the first section, what mechanism, and you learned mechanisms, could be used in moderating the energy charge. And I don't have a yes or no on this one, so just think about it. You've got a couple of minutes. You're a quiet bunch. Well, I forgot when we started, but so I can't do yes or no and count votes. So this time, if anyone wants to volunteer and raise your hand, I might call on you. Anyone have any thoughts about the types of mechanism? Yes? But how? Enzymes can be inhibited by different ways. But you see, I'm asking, enzyme inhibition is the answer, but you could have mixed, you could have competitive, you can have allosteric. No. No, I mean, non-competitive it isn't competitive, and it's not competitive because you're saying it's not necessarily involving the substrate, right? Okay, so it is non-competitive. But there's a particular type of non-competitive. Yeah, it's allosteric. Nobody else had a hand up to the viewing audience, so that's why we gave it all to our volunteer over here. It's allosteric. When you think about it, you can have, like for phosphofructokinase, for the control step of glycolysis, you could have obviously things happening at the site, which since ATP is a substrate there, anything that happens at the site would be competitive. But the way we get our adenyl nucleotide charge to be enforced in many ways is by AMP, ADP, or ATP binding to enzymes at non-active sites, and by doing that, changing the inhibition to something that's a non-competitive form. Now, we can't really measure this. We can't really measure it for everything and say that's driving totally energy charge, but we can see that we have allosteric effectors all through metabolism of adenylate nucleotides, which can be what's causing that. So that's our energy charge. Now, for this one, just to see here, coenzymes of intermediate metabolism. Now, Dr. Ponticelli was supposed to use pyruvate dehydrogenase as an enzyme, and I believe he did. And that introduces you here to a whole lot of different enzymes and cofactors that I need for metabolism. You can see that that's what we mean here when I say pyruvate dehydrogenase. So these you've already seen. You're going to come to know, in addition, pyridoxal phosphate and amino acid catabolism. When you see carboxylation, the cofactor that is involved mostly there is biotin, and these are vitamins, B vitamins. Reductive biosynthesis, we already talked about NADPH and NADH being involved a bit, and we also have this tetrahydrophosphate involved. Can't pronounce it, but it's one carbon metabolism. Coenzyme A you should be familiar with, and acyl carrier protein. They're both carrying functions. And again, in one carbon metabolism, which is a particular way of moving carbon groups that you'll get in the second portion of metabolism, you're going to have tetrahydrate folate, S-adenosine methionine, and this should have been a hydrotetrahydrofolate, so I didn't, one of them should have been without it, and methylcobalamin, vitamin B12. So as you go through this, what you need to do is think about vitamins, and from a nutrition point of view, think about cofactors. Because remember what I told you, that if you're missing one thing, the reaction doesn't occur. Why don't you want to open? So if you're missing a cofactor, you could have all the substrate, you can have all the enzyme, it's not going to occur. And that means that you can see why, if you're missing a particular vitamin, there might be a whole series of diseases that you get, or symptoms that you get, because that cofactor is involved in multiple different types of reactions. So as you classify things, there always is going to be some question asking what's going to happen metabolically in the absence of a particular vitamin. So again, the study of metabolism in biochemistry is a nice mesh with nutrition, because of vitamins and other things that are involved, and physiology because of the function and the work that you need. So we're now starting our Unit 2, glycolysis, and this section is anaerobic processes, and glycolysis takes place in the cytoplasm, which is anaerobic, and that's part A. Part B would be glycogen breakdown. So first I'll go over some carbohydrate chemistry and types of reaction that should be reviewed, but seems more over the years to be not. What's your overview of glycolysis? And it has two stages. We go from glucose to glyceraldehyde 3-phosphate, and this is ATP utilization, which might seem odd because we want to do the complete oxidation of glucose to CO2 and water, and we want to make ATP, but because of the chemistry, we have to actually put some in. And as I'm saying, the complete oxidation of glucose to CO2 and water, what we have in glycolysis is we're going from glucose to pyruvate. So we're on our way, but we're not there yet. In stage 2 of glycolysis, when we go from glyceraldehyde 3-phosphate to pyruvate, we generate ATP. So then, of course, we need to consider what happens to this pyruvate that we're generating. Different things happen under different conditions. And what's the regulation of glycolysis? So as I go through the lecture, you need to know what steps ATP are used and formed, which steps is NADH formed. Because we're going to have a problem where our adenine nucleotide metabolism is tied to what the NAD plus NADH that you have in the cytoplasm, because it's critical for glycolysis. But most of the NAD plus and NADHs involve oxidative phosphorylation, and that's all going on inside the mitochondria. So we have a lot of stuff in the mitochondria that would make our glycolysis go better, but the cell can't get it in the cytoplasm. So that's why it's important for the moment to just, we're not going to do the regeneration here, but to show where our NADH is formed. And that's, and I just described the effect of compartmentalization on glycolysis. Mostly that we have this problem here. What are the 3 catabolic fates of pyruvate? And how our control step of a pathway can be predicted from the delta G. Now, what I mean by that is that cell doesn't want to be inefficient. So if you think from a chemical point of view, as you're doing from one, reactant A to B to C to D, you reach a point where you're committed to glycolysis. So when you go from A to B, that B intermediate might be used in multiple pathways. But once you get to C, it's not able to do anything else but glycolysis. And not surprising when those pathways, which are the controlling steps, have a very high minus delta G. So some way you can look at something and see, gee, this reaction has the highest minus delta G under physiological conditions, so that's likely to be the control step. And also you'll find the control step likely to be the first committed step in the pathway. And phosphofructokinase 1 is the critical control step, satisfies these criterion in the regulation of glycolysis. I am not giving away anything when I say there will be at least one question on phosphofructokinase 1 on the exam. And if you're following and organizing things, this should become very clear to you that this is a major topic. Okay, carbohydrates. It's the most abundant form of energy on the planet. And they basically have a structure CH2O, and it just goes on. So the difference you can see that we're having the same structure here as our fats will, but we're missing nitrogen to be proteins. We think of them as sweet because they activate those receptors in many cases, but that doesn't mean they're all sweet. And they can provide a lot of conformations, and they're very interesting structural component in animals and plants, more so in plants when you see the carbohydrate being involved in structure. They are strongly antigenic, but very important as signaling molecules. And these are the carbohydrates that mark your identity via the HLA antigens, which isn't really part of this course, but you need to differentiate self from others and microbes, and it's carbohydrates that do it. This is the major food group for humans. Again, we're talking biochemistry, but carbohydrate is the same nutrition carbohydrate. People eat more carbohydrates than they do, well, they should, than fats and proteins. Monosaccharides. So the simple form of a carbohydrate, remember stage 1 of metabolism will be getting a polysaccharide to a monosaccharide, and that would be the monomer. And they basically, this would then be one sugar, and the ones that are important in metabolism to us are 3 to 7 carbon atoms. Usually they're colorless, water-soluble, crystalline, and the common examples that we use, obviously, glucose, fructose, lactose. So here's our monosaccharides. And you should be familiar with all these different ways of looking at a molecule. I can put up any one of them. This is the Fisher projection, which shows you a lot of how the chemistry is going on. When you want to emphasize the ring structure, we use Haworth projections. And then you could see whether the orientation around the bond, up or down, in the, here's a glucopyranoside. And you could also have a space-filling model or just a ring structure. So an example, we've got triose, tetrose, pentose, hexose, heptose. What our glucose and lactose are a major part of this course. For nucleic acids, we've got to get Dr. Tabo a lot of ribose to work with, so that's going to be important. And we have metabolites, glyceraldehyde we see a lot. The tetrose and the heptose are not involved a lot in this metabolism section, except they are intermediates in the pentose phosphate pathway. So our common disaccharides, we would have sucrose, which is what we'll put in our sugar, which we have glucose and fructose combination with a bond, a beta 1,2 bond, galactose, milk sugar, alpha 1,4, maltose, which is a glucose and glucose disaccharide, and here we've got an alpha 1,4, which you can see 1,4, and trehalose is a non-reducing sugar, and we don't see a lot of that. But you need to recognize these bonds, whether they're alpha 1,4s or, well, you don't have it up here, an alpha 1,6, and understand what that means. Because we're going to be building up polymers of glucose. That's what glycogen is. And so if everything can only be built alpha 1,4, you couldn't build up the polymer. But you can actually do an alpha 1,6 linkage here also. And that is chemically very different, and different enzymes work on it, but enables you to build up a polymer. But for the moment, just to understand that we have orientation around bonds and how our dimer is built. So our linkages, here's our maltose with our alpha 1,4, to just emphasize this again about the alpha 1,6. So here you see 1,4, here you see 1,6. So this molecule, which has an alpha 1,4, could also be linked alpha 1,6 somewhere. So this alpha 1,4 here, and this is your alpha 1 to alpha 6. So this little molecule here of glucose can be linked to one chain up here through an alpha 1,6 linkage, and another chain down here through one of these alpha 1,4 linkages. So you need to recognize as we go over this that there's different chemistry involved in alpha 1,4 and alpha 1,6. Now the glycosidic bond, again we have our maltose. I don't know if they still do it, but in grade school they used to do starch tests where you would react potatoes with things and they turn blue. Anybody see a blue potato somewhere in their education? Let the viewing audience say that there seems to be a 5% saying yes. That happens because of